I really like talking about malaria, and I hope that shows in this hour. So this is actually a neon sign. In addition to a disease, malaria is also the name of a rock band, though I haven't really heard their music. This was one of their neon signs. You don't find this sign in hospitals. So talking about malaria in Seattle is really special, I think, because, and I actually moved here to Seattle in large part because of the really great malaria research going on here. There's all these institutions, which you see at the top, who are working in one way or another on many of the aspects I'll talk about today. And we also have to acknowledge that the Gates Foundation has its home in Seattle and has made malaria one of its top priorities. So last year, just before I gave a different talk on malaria, there was an NPR story, and Kent Campbell, director of Seattle Biomed, which was at the time known as SBRI, was quoted as saying the greatest concentration of malaria research in the world is going on along the shores of Lake Union right here in Seattle. And so, be it drug discovery, diagnostics, vaccine development, there's a really wealth of research going on in this area, and it's neat to be a part of that. So I'll try to share some of that as we go along. So what is malaria? It seems like something very simple, seems like something that people talk about off the cuff, and it seems like it should be simple. It's a single-celled, malaria as a parasite is a single-celled eukaryotic organism. The asexual stage merozoite is shown there on the left, and it's transmitted by female Anopheline mosquitoes, shown here in a cartoon that Dr. Seuss did in World War II to tell troops to stay away from mosquitoes. And it causes the disease that we know of as malaria exclusively when the malaria parasites infect red cells, though as you'll come to find, they infect a lot of other cells as well. They just don't cause disease there. So simply put, this is the basic equation that gives you malaria, but it's much more complicated than that. And it's very important as well. So every day, 3,000 people die from malaria, and this is about 3,000 people gathered at a malaria control rally in Zambia, where they're lobbying for things like better government control of malaria, better distribution of bed nets as a right rather than a commodity, etc. So it's a lot of people, and although the toll of malaria is decreasing, we think that this year and last year we're just moving under 1 million deaths per year from malaria. It's still a staggering toll compared to most other diseases that we think of in the developed world. So there's the death toll. There's also the morbidity toll. There's over 500 million infections annually. These come from a variety of species, which we'll talk about. Most of these infections are not severe. They occur in adults, and they have flu-like symptoms. They're debilitating from the sense that people can't work, but they don't result in hospitalization. But a good proportion of them, 100 million or so, do result in hospitalization, and 1 million of them, and hopefully that number is decreasing, result in death. And death primarily occurs in children under the age of 5 living in sub-Saharan Africa. One of the things that economists and malariologists have come together on is that the map of malaria and the map of poverty in Africa really overlays one another. And there is great work that Jeffrey Sachs and others have done showing that the relationship between malaria and poverty and economic status is bidirectional. So if you eradicate malaria, you raise people's economic status. And by the same token, if you raise their economic status, you can help eliminate malaria. It's really a two-way street. And so it's neat to work on a disease where you think that a medical intervention could do more than just prevent disease. It could actually raise someone's economic status and improve their life in many different ways. So the overview for the talk today is that I'm going to talk about the history of malaria just briefly. I think it's interesting to kind of situate where we are now with where we've been. I'll show you a video of the malaria life cycle, which people seem to like. And then we'll talk about the disease, its complexities, the complexity of its pathophysiology. We'll talk about ways that groups around the world are combating malaria using drugs, trying to make vaccines, and also trying to prevent the mosquitoes from biting people, which if we could do that, we could eradicate malaria. But mosquitoes, as you know, are pretty difficult to eradicate. And then we'll talk as we go along. I'll try to point out some things which I think are good point of care diagnostic questions. And by addressing those questions, some of you in the room might be able to have a role in helping to control and eventually eradicate malaria. So we'll start with a history. So I sort of have broken up the history of malaria into 3 eras. There was the era where we knew nothing, there's the era where we knew mosquitoes were not a good thing, and there's the era that we're in now. And so Historically, you find reference to malaria and to fevers dating back into the 2700 BC in Chinese historical documents. At the time, it was known as a blackwater fever. So in Rome, where the swamps were, it was called a blackwater fever because, of course, the mosquitoes would develop in these stagnant pools of water. And some people thought that the ague monster, which is depicted here, was actually the bringer of malaria, and we came to find out that wasn't actually the case. The name malaria comes from the Latin malus area, which means bad air. And it's interesting that this name has been twisted in recent years into the name of a company called Sanaria, which is on the East Coast. Sanaria means clean air, and the goal of Sanaria, run by Steve Hoffman, is to develop a viable malaria vaccine from a sporozoite, which we'll talk about later. So the Latin is coming up again and again. In 95 BC, one of the first microbiologists, if you will, Lucretius, said that perhaps some kind of microbe, something very tiny, may cause swamp fever, but we know that it was about 1800 years before we had any sense of what microbes actually were. In the 1600s, Jesuit priests used cinchona bark to make tea, and the cinchona bark contained quinine, which was really the first drug that we ever had against malaria. And it's still a very good drug up until this day, though it has a great deal of side effects. So the second era of malaria was the recognition that there was a microbe involved, that it was vectored by mosquitoes, and that if you could prevent mosquito bites or kill mosquitoes, you could prevent malaria. So in 1880, Alphonse Laveran in Algeria showed that there was in fact a parasite in the blood of people who seemed to have these fevers. In 1897, Ronald Ross in the Indian subcontinent showed for the first time that a mosquito could vector this infectious disease. In 1939, Mueller developed a DDT, a drug which today we think about as a bad insecticide, but in fact this drug had tremendous impact on malaria throughout the world and was used in the World Health Organization campaign against malaria in the 50s and 60s. Ultimately, DDT was discontinued, and this campaign did not reach perhaps the most important parts of the world stricken with malaria, that is Africa. And so the campaign, although it made great strides in certain places, was ultimately not successful. Starting in the 1960s, we started to see the emergence of drug resistance, first to chloroquine, and as we'll talk later, to other drugs. And this has continued and will continue to be a problem for people developing drugs. So this era of malaria, I like to show this picture of the state of Illinois where I did my Ph.D. This is a 1920s era picture from a public health agency showing the distribution of malaria deaths in Illinois, which we all think of as a malaria-free region today, which it is. But you can see there's a really nice north to south distribution. So the darker the squares, the more deaths there are in malaria. And in the south, there are more deaths. And up in Cook County, where Chicago is, there's very few deaths. So malaria today is thought of as an African, an Asian, South American issue. But we have the vectors in the United States. And in the 1930s, we had 1 million cases in the United States. And it's this recognition that mosquitoes and malaria go together that made the United States malaria-free as it is today. So the current era of malaria is really typified by a move into the molecular era. We've sequenced the mosquito, the human, and multiple species of the parasite. And essentially the world today is malaria-free in the developed world. So most of Europe, most of the United States. Although climate change threatens to change that, we have lots of public health infrastructures that deal with things like mosquitoes that lead to urgent and prompt and complete treatment of patients here as long as the disease is recognized. In the developing world, it's a very different story. And if you think about population growth, some people will actually say that the number of people who are at risk for malaria is on the rise, even though we know more and more about it and we have more and more drugs. The reasons are that drug resistance is increasing, insecticide resistance is increasing, and this is a disease where the perfect drug is less than a dime a day or even a penny a day. And so there's not very much incentive for pharmaceutical companies. So groups like the Gates Foundation and governments have been trying to change that to incentivize the investment into drugs and vaccines. But it's nothing like the kinds of drug market that we have here in the United States. And then, as I mentioned, climate change. So this is a picture from a malaria mapping group that is based in South Africa, which shows the relative risk of malaria throughout the world. And you can see that the most intense malaria transmission of Plasmodium falciparum, which is shown here, is in Africa. So the other historical context to put malaria in is how it has affected human evolution. And we're not going to really talk about it except to say that this is a whole laundry list of diseases and human genetic traits which owe their existence in one way or another to the selective pressure put on by malaria. So sickle trait, for instance, is thought to in some way protect against malaria, and yet we know that if you inherit 2 copies of this defective gene, you have a very bad phenotype, and without modern medical treatment, those patients with sickle cell anemia died very young. So the selective pressure of this disease over the years must have been tremendous in order to make it a viable option to inherit such a disease-prone gene. And so many authors have said that malaria has been the greatest selective pressure on the human genome in recent history. So malaria is a disease, the parasite, the life cycle of malaria involves 2 hosts, mosquito and human, and it's sort of a life cycle of cycles. Within each host there's a cycle, and so we'll look at that. But basically all of the species undergo very similar life cycles. So up until a few years ago, we used to think of malaria being caused by 4 species, but in fact, there's at least 5 species of parasite that cause human infections. Plasmodium falciparum causes the most deaths. Plasmodium vivax probably causes the most cases of what's called benign malaria. And then Plasmodium ovale malariae are much less common and not as severe. But a newcomer to the group is Plasmodium knowlesi, which used to be thought to be transmitted exclusively in non-human primates in Malaysia and areas around there, but more recently has been shown to cause severe P. falciparum-like disease. And so it may be that there are other species of malaria that we don't know about that are capable of infecting humans. But for your purposes, I think this is the inclusive list at the moment. So this is a life cycle video of malaria that came from Steve Swank, a medical illustrator. And this is the female Anopheline mosquito who would usually be flying at night, homing in on a person here. So she bites the person, and if the mosquito is infected, she takes a blood meal, and while she takes a blood meal, she injects these purple motile sporozoite parasites. These are a sexual form of the parasite, which homes from the bloodstream into the liver, where it infects a hepatocyte, and over the course of about a week grows to make 40,000 asexual stage merozoites, which then bud from the hepatocyte. And at this time, we don't know that you're infected with malaria. So the merozoite is special in that this is the infectious form, so it buds out of this little merosome. This is the infectious form shown here that is capable of infecting red cells. So it attaches and invades the red cell by invaginating the membrane and forming a vacuole. And inside this vacuole, it grows up, in falciparum's case, over the next 48 hours, first to a ring stage parasite shown here, and eventually to a more mature parasite known as a trophozoite, a growing form, and eventually a schizont. So during this time, this is the time where we actually know that we have malaria. And this classically gives fevers, chills, myalgias, pain, headache, etc., but can also cause very severe disease, including cerebral malaria with severe anemia, etc. So the parasite grows up and then it divides to form a schizont. Now there's 32 of those little merozoites inside, and it adheres to the cell wall, so it evades immune detection. Eventually, after about 48 hours, it lyses, releasing more of these merozoites, which then go on to propagate the cycle again and again. Eventually, if you get enough parasites, you get what are called gametocytes, which are sexual stages of the parasite. There's a male and female, and if a mosquito comes by at the right time, she takes a blood meal, and the blood meal contains these gametocytes, which do nothing in the human, but carry the future of the parasite forward in the mosquito. So the male gametocyte changes here to form gametes that fertilize the female to form a zygote. The zygote then implants in the cell wall, in the gut wall, forms an ookinete, which in a temperature-dependent process develops into an oocyst, and the oocyst contains these sporozoites that started the whole process off. Eventually, the oocyst ruptures, and it releases the sporozoites, which amazingly home to the salivary glands of the mosquito, which are ready to then propagate another cycle. So it's a really interesting life cycle. I think it's much more interesting when it's presented in this form than on a single slide, and it's very complicated. So needless to say, or maybe not, the only form that we can detect in humans is the parasite as it exists in the red cell, which is already too late to know whether or not the person was infected in the liver. the only way you could get a liver diagnosis would be to take a liver biopsy in a person who was otherwise well. So we wouldn't do that. We do that to mice, but we wouldn't do that to people. And so by the time you diagnose the infection, it's already been going on for quite a while. So the disease, which is a complication of all of the red cell stage of infection, can go a number of different ways. So people can have acute disease with fevers, chills, myalgias, etc. And it can be uncomplicated, meaning some people don't even notice the fevers or chills, or they don't manifest, maybe because they've been infected again and again over their lifetime, and so now they have enough baseline immunity to prevent any manifestations of disease. Or they can be severe. People can develop anemia because so many cells are parasitized, and in addition, the infection can shut down the bone marrow in one way or another, so that your anemia can be much greater than the number of cells that are parasitized. The sort of harbingers for very severe disease include hypoglycemia, severe anemia, when people go into respiratory distress, or perhaps the most feared complication of all, which is called cerebral malaria. People lapse into coma and maybe never come out. But the amazing thing is that children, I've seen this in Malawi, children will be in cerebral malaria, they'll be treated, and 2 days later they'll be running around the hospital ward. So it's very unlike a lot of diseases where you see someone in coma and you don't expect them to come out. With drug treatment, many of these complications are reversible. Sometimes they're completely reversible, and sometimes people, particularly who have cerebral malaria, succumb to a variety of sequelae, which include blindness, deafness. People, when they're children who have this, they have learning disabilities that are then lifelong disabilities that the whole society has to deal with. And then, of course, many people die. People can also have chronic infection. Chronic infection, for instance, in a pregnant woman, most notably can lead to low birth weight babies, and low birth weight babies are then subsequently at a risk of a million other things. And so the impact of malaria, there's a very much a sort of splinter effect. It leads to lots of other problems as well. So not just medical problems, but the parasite itself can complicate the disease. So it could be drug resistant. There could be more than one strain of parasite infecting a person. There could be more than one species infecting a person. There is one amazing report of 4 different species of malaria infecting a person, though I don't think anybody has to really expect that. The host can be semi-immune, can be completely naive. If most of us who are from the United States were to travel to Africa, we would fall into that completely naive category and be at risk for severe malaria, much more so than someone who was born and raised in equatorial Africa and has had malaria, whether they know it or not, again and again. And then, of course, environmental factors. So places with a lot of rainfall where malaria is endemic have a pretty stable level of transmission, but places with epidemic malaria where it comes and goes, people's immunity can wax and wane, and those people are sometimes at greater risk from disease than people who live in endemic regions who are sort of continuously being primed by the parasite. So after you get bitten by a mosquito, you have about 7 to 30 days, depending on the species, before the parasites emerge from your liver and start infecting your red cells. And at this time, you don't know you have malaria. Nobody knows you have malaria. The incubation period is longer with certain species, and there are certain forms of the parasite known as hypnozoites, which are these dormant forms of the parasite that are formed in P. vivax and P. ovale infections that may lay dormant in your liver for years and years. There's stories of people 30, 40 years after leaving an endemic area, something happens to them, they get the flu, something changes in their immune repertoire, and suddenly these hypnozoites emerge and they have malaria, but they haven't been in Africa for 30, 40 years. Most of these make good case studies, but hypnozoites are a significant problem that needs to be dealt with because we have no way of diagnosing that they're there. So that's actually a really interesting diagnostic issue. Anyway, this incubation period can cause a lot of missed or delayed diagnosis. So uncomplicated malaria, as I mentioned, fevers, chills, headaches, muscle pain, nausea, vomiting. You typically see jaundice with Plasmodium falciparum because it can infect a wide range of red cells, young red cells, old red cells, etc. People get large livers and spleens as those infected cells are filtered out. You can sort of try to decide what species you're infected by if you follow the periodicity of the attack. So when you're first infected, there's very few parasites. But eventually, one of the parasites that emerged from your liver, one of those packets of 40,000 merozoites, will become 400,000 and then 4 million, and one of those will take over and synchronize your infection. And so for most infections, over the years we've recognized that the periodicity either comes every day for P. knowlesi, every 2 days for Falciparum vivax and ovale, or every 3 days for P. malariae. And over the years, these have become known as tertian and quartan malarias for coming on the 3rd and 4th day, fevers coming on the 3rd and 4th day. But I'll tell you that we don't see this as much as you would think. So the classic stages of malaria are classic but uncommon. And presumptive diagnosis, which is used in people presenting in a equatorial region with fevers, is often used and is a quite sensitive approach to detecting malaria, meaning if you have malaria and you come in with a fever and we treat you, that's great. But you may well have something else. You may have salmonella, you may have the flu, you may have a number of other things that we would miss. So presumptive clinical-based diagnosis is very sensitive but not very specific. And these are the classic stages that are associated with synchronous infection. So people get cold just before the parasites rupture, then they get fevers as the invasion is going on, and then eventually they get sweats. This all occurs over 6 to 10 hours, and then if you have one of these kind of classic presentations, it occurs again either 24, 48, or 72 hours later. So severe malaria is primarily the domain of Plasmodium falciparum, although increasingly we've recognized that Plasmodium knowlesi can also cause severe malaria. And this mostly occurs in children, it occurs in pregnant women, it occurs in travelers, people who have not seen parasites again and again in their life. And all these complications listed here require aggressive treatment. And some of them, for instance, trying to decide whether someone has cerebral malaria or not would make a really good diagnostic project, because it's not as simple as the parasite just being in the brain. And so people have looked at cytokines and other markers to decide is there some kind of cytokine storm going on that puts these people into coma. We really don't know, and there's even some cases where we look in the brain, we've decided they have cerebral malaria, the person dies, we look in the brain and there's no parasites in the brain. So it's a very complicated pathophysiology. But all of these conditions require prompt intravenous treatment with drugs. So then there's also this issue of malaria relapses, which I've already touched on. There are 2 species, Plasmodium vivax and ovale, that if incompletely treated, that is, if treated with the drugs that are perfect for falciparum, hypnozoites can form for these other species, and they can come back months later. So if you're in the business of eradicating malaria, you need to address this hypnozoite problem as well. And there's only one drug that kills tissue schizonts, as hypnozoites are known, and that's primaquine. And so this is a PubMed shot of a case where someone relapsed 36 years after their original infection, and this is not very uncommon. This is a picture showing what the hypnozoites look like in the liver if you were able to get a piece of liver. So I don't know if you looked, I handed out this paper about when is malaria malaria, and some of it I alluded to earlier. We have malaria the parasite, which we should actually call malaria parasites. We have malaria the disease, which is what clinicians refer to, and we have malaria, which is what policy and public health people refer to when they're trying to dampen down the overall burden of disease. But there's also when is malaria malaria diagnostically, and you can be infected but not diseased, and so this is a great distinction that should be made. Clearly, people who have, let's go back here, people who have 250,000 parasites per microliter of blood are really sick. They might well die, and they require treatment. It's not very hard to diagnose these people. But as the parasitemia decreases, so to 10,000 parasites per microliter, may be the threshold at which someone who is immune, who's had a lot of malaria in their life, their immunity may no longer control this. None of these are hard and fast numbers. I would like to also point that out. None of these are hard and fast numbers. So one of the rapid tests that's out there, the Binax Now test, has a sensitivity of 5,000 parasites per microliter, which isn't very sensitive compared to some of the other tests I'll talk about. So then if you're a traveler, you have a lower threshold for disease, perhaps 1,000 parasites per microliter, but maybe even 100, depending on just who you are. The 2 tests that I think are most useful to think about in the future are thick blood smears, which have been the standard for looking for malaria throughout the world, which have a sensitivity of about 5 to 20 parasites per microliter, and then molecular testing, which is a domain which I've become involved in, which has a much improved, perhaps 1,000 times better sensitivity, down to as little as 20 parasites per milliliter, not per microliter, but per milliliter. And the question with a test that's this sensitive, if you test someone from a malarious region, and they have a fever is if they have a parasitemia that low, does that have anything to do with their fever, or do they have something completely different? And we can't answer that with a single test. We have to take everything into account. But I'll show you the value of what I think molecular testing is. So this is a graph from a vaccine study that was performed in the Netherlands, where they infected people with sporozoites. They actually taped a cup with mosquitoes on their arm and let the 5 mosquitoes bite them, and then they waited a week, and eventually the people became parasitemic. We recently did a study like this at the Seattle Biomedical Research Institute as well, but the data isn't ready to show, so I'm showing this data, which is very comparable. Basically, in the vaccine arm, let me see if I can get the pointer going here. In the vaccine arm, nobody developed parasitemia, but in the control arm, which is what I'm sort of more interested in showing you, starting on about day 6 after infection, the parasites left the liver, they started to infect the red cells. They went up, they go down because the mature forms of the parasites sequester in the blood vessels. Then 2 days later, they go up again, they go down, they go up. The problem is that all of these points that are not in the red box would not have been detected by anything other than molecular testing. And so for something like a vaccine study, which is what we've become involved in, and have a test with this kind of sensitivity, a molecular test is the only way to go. It's the only thing that gives you really good informative data. Otherwise, all you get is the very peaks, or you have to wait until the person is really sick. And for vaccine studies, we don't want to put someone at that kind of risk. But the question in the field would become, if you had a test that was this sensitive, I keep losing the pointer, if you had a test that was this sensitive, what meaning do you give that from a clinical point of view? Because in many cases, our tests were, in the most recent study, were positive 4 days before the person became symptomatic. So it'd be great if we could treat them and avoid all symptoms. But in someone who's presenting with fever, we worry that maybe they run along with that many parasites in them if they live in Africa, and this fever is due to something completely different. So we'll talk a little bit about how we're combating malaria using this sort of 3-prong approach of vector-controlled drugs and vaccines. So first, vector control. You would think that mosquitoes should be very easy to eradicate, given this cartoon here. So this is says, Morty, what are you doing? That's the bladder, and Morty is looking like a really engorged mosquito here because he's not taking up blood, he's tapped into the bladder. So we think they're not very smart insects, but in fact they're very pervasive, and we have to have mosquito control as one of the prongs of malaria control. But it's more of an economic and political issue, at least using basic public health interventions, and often those commitments for the long haul are lacking. So it doesn't really help a community to give them 10 years worth of malaria control and then have that fall off, because then you've left a community basically naive who hasn't had malaria for 10 years, and then they all look like us, all a bunch of travelers, except they live there, and now the malaria could come back rip-roaring. So mosquito control is really important, and the main approaches for mosquito control have, as you may have read in the news, insecticide-treated bed nets. These are really great if you sleep under them. If any of you have ever slept under a bed net, it's not the most comfortable, particularly if it's really hot and humid out. So they do reduce all-cause mortality by about 20%. And there's ongoing debate whether bed nets should be a commodity, should they be a right, which would be great, or, should they be a right, which would be great, or are they a commodity? And in many places, they end up being a commodity, even though that wasn't the initial intention. So in addition, DDT and other insecticides are used very differently than DDT was used decades ago to do indoor residual spraying, where like the amount of DDT that was used on a plantation decades ago is now enough to treat thousands of homes. We also do things like try to reduce larvae in standing water. And there are efforts underway, although they're somewhat controversial, to make mosquitoes that are somehow incapable of carrying malaria, but are more fit than wild-type mosquitoes, to put them out there and then have them take over the niche of existing mosquitoes. As you can imagine, this sort of genetic engineering comes with a lot of ethical issues that need to be dealt with. So in terms of drugs and resistance, drug resistance is a big deal. And this is a case study from 1997 in the New England Journal of Medicine showing that there was this person who was treated repeatedly but got malaria 13 times because they basically didn't have the right mix of drugs because they didn't know what was going on with the parasite that this person had. So resistance has become the main issue in malaria drug development. And you want to make a drug that's new, that the parasite has never seen before, but you sort of realize that as a eukaryotic organism with thousands of genes and a lot of biochemical pathways at its disposal, this parasite will more than likely outsmart, if you will, most drugs that we throw at it eventually. So we've developed things like combination drug therapy, et cetera. The same principles that go into bacteriology are now being applied in malaria. And the principles of prescribing basically deal with what species do you have. So the issue there is if you have Vivax or Avala, you need to add primaquine to whatever your treatment is. How bad off are you? Do you have uncomplicated disease? If you do, we can give you oral drugs. But if you're in a coma, we obviously have to give you IV drugs. And where are you? So oftentimes we know you have malaria, but we don't actually know what species you have. And maybe the thick smear isn't even indicative of this. But because you're in South America, or you're in India, or you're in the Gambia, we can guess with pretty good certainty what species you probably have. And so these are the drugs, and basically most of them work here to kill parasites in the blood stage, and the only drug that kills in the liver is primaquine. As you can see, all of these little stars up here indicate known drug resistance, which we'll address in a second. So these are the various classes of drugs. You have quinine, you have the antifolates, artemisinin, antibiotics, which are a very common drug taken by travelers. So people will take doxycycline when they go abroad. Alternatives for travelers include Malarone and mefloquine, though mefloquine has been famously in the news as causing neuropsychiatric disturbances, and although it's great in that you only have to take it once a week, this has made many people turn to Malarone or to doxycycline. The issue with doxycycline in someone who's going to equatorial anywhere is that it causes you to be sun sensitive, and so this is again a blessing and a curse for in terms of traveler-related medicine. And then atovaquone, proguanil. These are sort of the main categories of drugs. And the newcomer on the block is artemisinin, which is derived from a plant which was first discovered as a Chinese herbal remedy. And they all have different mechanisms of action, as we would suspect. And they, for the most part, all have had resistance arise in them. The only one which hasn't is doxycycline. So unfortunately, even artemisinin, which is the newest drug on the block, has had reports of resistance in parts of Asia. And so this is a map showing the resistance to chloroquine. So chloroquine resistance is very widespread, especially in Africa. In fact, the only places that really anybody uses chloroquine anymore is in the Caribbean. And then there is also resistance to sulfadoxine pyrimethamine, to mefloquine, which is our drug that was developed by the Army and used once a week for treatment. Most of mefloquine resistance has been restricted to Asia. And then there are, in white is obviously all the malaria-free areas. So we'll add that artemisinin resistance has cropped up here, probably because the drug was sold illegally on black markets before it was adopted by the World Health Organization. And so it's not just the illegal sale, but it's the improper prescribing of it. Artemisinin is now being marketed as a combination, so that you have multiple drugs acting in multiple different mechanisms of action, and you have much more likelihood of complete killing. Because if you don't have complete killing, you basically bathe the parasite in this drug and really provided and enriched for selection for a resistant parasite. So the way we are monitoring for drug resistance is we can treat a patient and see if they get better. It's called a therapeutic efficacy test. If you knew that you had a resistant parasite, you probably wouldn't want to go down this road if you somehow knew magically. So then we also have in vitro sensitivity assays where we grow parasites in various dilutions of drugs, but this takes a while. This is really not the best diagnostic test, though if you're a public health entity and you want to understand the distribution of parasites in your country, this test works fine. And then molecular markers. So as you might imagine, there are PCR and other based tests to look for resistance. And this table shows the various genetic determinants of resistance that have been discovered in Plasmodium falciparum and confer resistance against chloroquine, mefloquine, sulfadoxine, pyrimethamine, and atovaquone. And so each of these genes could be probed with a gene-specific molecular test to show whether the predominant parasite in a person was resistant or not. And this is a very good area, I think, for point-of-care diagnostics, although it would have to be tailored to the kind of resistance that is known to be wherever the test that is under development would ultimately be used. As I alluded to before, artemisinin-based combination therapies have been rapidly adopted by the World Health Organization, and this shows the shift to artemisinin-based therapy in green from other first-line treatments between 2003 and 2007. So the World Health Organization is completely on board with artemisinin-based therapies, and this is having a great effect and probably has something to do with how we've pushed the malaria death toll to under 1 million. In the past, it's been routinely quoted over the last 10 years that the death toll was between 1.5 and 2.7 million deaths per year. And so getting it under 1 million, although still staggering, is quite an achievement. And then this alludes to the artemisinin resistance, which I've already mentioned, which has emerged on the Thai-Cambodian border. And there is one report in the New England Journal showing in vitro drug testing with decreased sensitivity to artemisinin. And they think that it is not due to the 2 genes listed here. So my area of interest has become immunity to malaria because I feel that drugs are a very important part of our arsenal against malaria, but the only, in my mind, true way to eradicate the disease is to give your immune system some endogenous protection against the parasite that I think could ultimately lead to eradication of the disease. The problem is that there are no vaccines for parasitic diseases in humans. There are 36 vaccines out there, and they're all for viruses or bacteria, which arguably are much more simple, at least from a genomic structure, than this eukaryotic organism. In fact, there's very few antiparasitic diseases even in the veterinary world. I think I counted 1. So why is that? And the first question is anybody actually even immune to malaria? And there's 2 points to be made here. One is that if you live in a malarious endogenous region and you survive to the age of 5, then you have acquired, in most cases, natural immunity that prevents disease but not infection. So the immunity that you have is focused on the red cell stage of infection. Your liver still gets infected routinely because you're getting bitten by mosquitoes routinely, but the infection in your red cells is controlled primarily by antibodies such that you don't actually get sick, you don't develop coma, because all of that hopefully is behind you as a child. What's more important for vaccine developers is that we can also induce a kind of immunity. And the beauty of this induced immunity is that we think if we use liver stage antigens, we can completely protect against infection. So there's been basically 3 classes of vaccines that have been in development. And those are overlaid here onto a schematic of the life cycle. So I'll take you through this real quick. Mosquito bites the person, liver gets infected, 7 days later, parasites leave the liver, go into the red cells, cycle in the red cells, eventually make those sexual stage gametocytes, which go back into the mosquito. So the first class of vaccines are what are called pre-erythrocytic vaccines, and I think these hold the greatest promise to eradicate malaria. These are designed to attack either the sporozoite, to elicit immunity that either attacks the sporozoite or attacks infected liver cells. The reason infected liver cells are a great target is because they display MHC class 1 molecules, and so they display parasite antigens on their surface. Red cells, which form the target of the second type of blood stage vaccine, they don't have any MHC molecules, and so they have nothing to they don't process antigens, they don't have nuclei. They are these hemoglobin capsules that are perfect targets for malaria, but they have no way of showing the immune system that they're in fact infected. And so because of that, T cells are really not effective against infected red cells. And then eventually here, you have a third stage of vaccine, which is called transmission blocking vaccines. And the hope of this group is that if we could make an antibody against a protein which wasn't expressed in the person but was critical once the parasite went back into the mosquito, well, when the mosquito takes a blood meal, they take up your blood cells, they take up infected mosquito or infected red cells, and they take up whatever antibodies are in the mix as well. And if that antibody could then act in the mosquito rather than you, we could actually interrupt transmission in a really remarkable way. So maybe what we actually need is all 3 vaccines. And not only is there the issue of which type of part of the life cycle to address, but there is what is the best delivery approach. So the best way that we know so far to completely prevent infection is to take this motile sporozoite form out of mosquito salivary glands, to manually dissect them out of salivary glands, or to take the whole mosquito and irradiate the mosquito. So you either take the dissected sporozoites or the whole mosquito and you irradiate it lightly, enough to attenuate the parasite, but not enough to kill it completely. And then you either let the mosquito feed or you inject these attenuated sporozoites. And the immunity that's elicited there tends to be focused on the liver. And when you re-challenge someone sometime later with wild-type malaria, in most cases we've been able to achieve complete sterilizing protection. The problem is that making these whole attenuated organisms, which is the focus of that company scenario which I mentioned before, is not the most straightforward. We cannot culture sporozoites in a dish, and so we have to grow them in mosquitoes. In order to get them, we need to have the whole life cycle. The approach for most other vaccine developers has been to take one or two proteins and make what's called a subunit vaccine. The problem is that we have thousands of genes in malaria and so for thousands of proteins, and what protein do we pick? So we've picked a few over the years, circumsporozoite protein being the major protein. But it's likely that really great immunity, like that elicited by the attenuated whole organism, is dependent on much more than just one or two targeted proteins. That's actually the area I've become interested in, is to how do we define a signature, if you will, of all the T cells that have responded in someone who's immune, and how do we fish out what are the critical targets, be it 5 or 10 or 50 different targets. So the only vaccine that's nearing licensing is known as the RTSS vaccine, which consists of circumsporozoite protein fused to hepatitis B surface antigen. And the circumsporozoite protein is on the surface of the sporozoite, and so they've taken that, fused it to this other immunogenic antigen, and used it to immunize kids and other groups. And in kids in Africa, they found in a phase 3 study, this is GlaxoSmithKline, that it conferred 56% protection against disease at 18 months of follow-up. And so this is the vaccine that will go into licensing first and first be used in Africa. And 56% is great. If we could cut malaria deaths from 1 million to 500,000, that would be tremendous. But we think there's a great future in looking for other malaria vaccine candidates, be it a whole organism or some kind of complicated, very effective subunit vaccine. So the way forward in Seattle has been to develop what's called the Malaria Human Challenge Center, or Malaria Clinical Trials Center, where we have become the 4th institute in the world doing in vivo challenges in humans. And so what that means is we take volunteers and we are evaluating different vaccine candidates, be they subunit or whole organism. And so people are randomized into control arm or vaccine arm, and then they are challenged with, in most cases, 5 mosquitoes and followed throughout that whole course to see whether they develop immunity or not. This has, we've undergone the first demonstration trial, and we hope that the Human Challenge Center will be used for many other vaccine candidates moving forward. What this allows is, it allows us to have volunteers who've never been exposed to malaria and who in essence are adults capable of consenting to the whole clinical trial process. But because they've never seen malaria, they're basically immunologically naive, and in one way they're similar to children in Africa, the most vulnerable population out there. And so by studying their immune responses in great detail, we hope that we can do things like decide what are the most important antigens and maybe think about ways to formulate those antigens into a more simple vaccine that could be produced en masse throughout the world. So what's your role in all of this? First of all, I'd like to say we definitely have to do something about, we have to do more, of course. So the red line shows malaria mortality, which was growing at the current level of intervention. And it's growing because of population growth. Population growth puts more people at risk. Climate change puts more people at risk. If we scale up as has been proposed, and this is obviously a little dated because there's a mortality scale up by 2010 shown here. If we scale up, we can cut that mortality dramatically, and hopefully we can cut it to less than 500,000 in just a few years, and one day perhaps eradicate the disease altogether. I've heard that the eradication goal, which people feel is a reasonable goal, is the year 2050. And there's obviously been very few diseases which have been eradicated most of those have been vectored exclusively by humans. And so it is quite a daunting task to think about eradicating a disease which is harbored in a pervasive insect for much of its life cycle. But in Africa, it would really make a tremendous impact to eradicate this disease once and for all. So we hope that actually we are on that track. And as one indicator of this, I showed this last year too, interest in malaria and publications on malaria have been on the upswing for years now. The field has been stimulated by investment from government and from the Bill and Melinda Gates Foundation and other agencies as well. And so we hope that this increase in publications, increase in research results in some concrete change which can impact morbidity and mortality. As far as diagnostic and point of care challenges, I try to point out a few things as we've gone along, but I think in decreasing order of importance, these are sort of some areas to think about. So field tests are of course important, and the field tests that are out there either require an operator who's very skilled, that is a thick smear, or a point of care test like the Binax test, which all things considered is not as sensitive as many of the other assays that we have out there. The other issue in definitive field testing is trying to speciate parasites. Most tests now will tell you that it's falciparum or not, and that may be enough in some settings, but not enough if you're trying to eradicate hypnozoites and need to know if it's P. vivax or P. ovale. Perhaps as important is trying to know whether the parasite that's infecting this patient is drug resistant or not, so that clinicians can immediately guide their therapy. That not only helps the patient, but it also helps to prevent the emergence of future drug resistance. We need to better understand the mosquito vector, its resistance to insecticides. There's a great deal of effort underway to understand how the environment impacts this, so how soil, water content, and other predictors can tell you that a malaria epidemic is likely to occur, et cetera. The area that I'm interested in is correlates of immunity. So this is deciding on and defining tests that can take the blood from a person who we've declared as immune and try to ferret out what are the important immune responses that have made this person immune, that makes them different from someone who's naive. And obviously we have to do that in lots of people to define common immune responses, and those immune responses will vary depending on someone's HLA type. And so it's likely to be tailored to the community or to the country that we're looking in. As I've mentioned, it's important if we want to eradicate the parasite that we be able to diagnose hypnozoite infections. How we will do that, since this parasite is dormant and sequestered away in the liver, I don't know. But perhaps there's something that's shed by the parasite into the bloodstream, even though it's, quote, dormant, that could be detected by some type of future assay. And the other issue, which I haven't really touched on except for here, is there's a lot of drugs available in various markets, and they're not all good. And it would be really useful to be able to quickly help buyers of those drugs, be they individuals, but more likely the governments, know whether the drug they got is bona fide good drug or whether it's substandard or whether there's some issue that will make the drug less effective. I'd just like to acknowledge a few different people who I've worked with over the years. Kasturi Haldar, Narla Mohandas, and Joel Bremen are all malariologists, as is Terry Taylor. I've been working with Pat Duffy, who was at SBRI and is now at the NIH, as well as other colleagues at Seattle Biomed on all of these clinical trials, and we look forward to doing more of this work with them and to eventually publishing on this. And then, of course, the video, which was really great, came to us from Steve Swank, a medical illustrator who's given me permission to use this for academic purposes. So I think that combating malaria will ultimately save lots of lives, resources, and do a lot to change the world, and I hope that some of you can play a part in that in one way or another. Thanks for listening.